Hi everybody, welcome back. We're in the second week now, and I want to thank you guys for telling me a little bit more about you and the infographics. Really enjoyed that. You guys are doing some amazing things. You're amazing humans. And thank you for joining in on the live Zoom meeting and telling me your interests as well and asking questions. That was fantastic. But I want to continue on with our learning journey. And where we are this week is we're learning about acids and bases, the pH scale, and macromolecules. So let's get started. So, what are acids and bases? Essentially, when we look at a water molecule, H2O, remember that formula, H2 means that there's two hydrogens, and O means there's one oxygen. So H2O is telling you exactly what the atom, the molecule looks like, right? When we look at water, H2O, if we were to split off the hydrogen, one hydrogen, that hydrogen is now what's called an ionized hydrogen molecule. So I'm going to point your attention to the PowerPoint. And hopefully you can see my mouse here. I will go ahead and ping it so that it shows up a little bit better. And this ionized hydrogen molecule is now ionized because when it's pulled off of water, the electron, the one electron that it had, stays with the hydroxide, the OH on the other side. And so now it's ionized. It's a positive ion. It doesn't have its electron, right? And we also have what's called hydroxide, which is the OH, and it has now an extra electron. And so that makes it a little bit more negative. And it's also ionized, right? So when we look at these two things, this hydrogen ion and also the hydroxide ions, acids are fluids that have a greater proportion of hydrogen ions. That's a definition of an acid. And a base, they're fluids that have a greater proportion of hydroxide ions, OH, negative. That's what a base is. And acids have certain properties. Number one, they're very reactive, as you can imagine. They're missing an electron, so they want to react with any molecule that they can in order to obtain an electron. And so that makes them very reactive. Acids are also corrosive to metals. Acids can break down food in your digestive tract. We actually use acids, the acid in your stomach. And acids have the characteristic of being generally sour in taste. And you're familiar with this when you drink lemonade or have lemon juice, that's acidic, or vinegar, that's acidic, right? So you're familiar with actually the taste of acids. There's certain acids that you can't actually even think about putting in your mouth because they are so reactive, they would completely destroy your tongue or any tissues that they touch. Let's look at bases. So we talked about bases being hydroxide ions. They're the OH. And they also have certain properties. So strong bases are very caustic to your skin. So when I say that acids are reactive, you kind of know that because you're familiar with acids and you know that acids can actually cause you harm, right? But strong bases can cause you harm as well. And they're very caustic, burning to your skin. Bases can be found in many household cleaners. Bleach is a very common base that you're probably familiar with. And bases are generally bitter in taste and also slippery. So they have properties as well. But we have this scale that's been developed so that we can communicate with each other as to the properties of different fluids and different chemicals. we can communicate based on this number scale, which is a logarithmic scale, and I'll explain that in a bit. So if I tell you that something has a pH of 2, then you would instantly know that that's an acid. If I tell you something has a pH of 10, you would instantly know that that's a base because of this pH scale that we've developed. And so, so let me tell you a little bit about the pH scale. So water, H2O, pure H2O, has no other minerals or anything else in it. It's just H2O. It has a pH of 7. That's neutral on the scale. So 7 is neutral. Anything less than 7 is an acid. Anything greater than 7 is a base. Now, let's take a look at some common items that you're probably familiar with here on the pH scale. We have battery acid, which is here much lower on the scale. Here we have water in the middle at 7. And then we go 7, 6, 5, 4, 3, 2, 1. So you can see battery acid is a very strong acid. Soda is also very acidic, and it can be very surprising as to the pH of soda and how acidic it actually is when you look at it on the pH scale. Beer is also acidic. Coffee is also acidic. Now that takes us to water, which is our neutral pH, 7.0. And now let's start looking at some common bases that we're familiar with. Blood tends to be just a little bit more basic, slightly basic. It is not 7.0. It is usually about 7.3. Baking soda, as you can see, is 7, 8, about a pH of 9. Ammonia is an even stronger base. And of course, we have bleach here on the far end of the pH scale. Now, I said originally that this is a logarithmic scale. So it's not 1-to-1, meaning that when I look at water, which has pH of 7.0, which is neutral, and then I look at, say, coffee, which is 7, 6, pH of 5, the difference between each step on the pH scale is actually 10 times. So when I go from 7 to 6, that is a 10 times difference in acidity. Or if I go from a pH of 7 to a pH of 8, that's 10 times more basic. So that's an important point that I want you to remember, that the pH scale is logarithmic, and each increment on the scale is 10 times as acidic or 10 times as basic as the next increment. Okay? Extremely important. So if I were to go from a pH of 7, here from pH 7, 6, 5, how many times more acidic would coffee be as compared to pH 7 of water? Coffee is a pH of 5. So I want you to think about that question for a minute. So remember that we have a 10 times difference between each step. So if I start with a pH of 7, it's 10 times down to 6. And then another 10 times down to 5. So that means that coffee is 100 times more acidic than water. Okay, 10 times 10. Okay? So remember that because it's probably going to show up on a test question. All right, that's the logarithmic scale. But let's talk about what buffers are, because buffers are very important. Buffers in blood actually stabilize pH. And there's a great video that I think I'm going to find and put up for you on buffers and how they work, but let me see if I can explain it here. So let's talk about buffers in blood. So when your blood pH becomes too acidic, all right, so let's take a look here at the scale. Let me see if I can get my mouse to actually point up for you. We're here on the left side. When the blood pH becomes too acidic, here's 7 in the middle. This is giving you sort of the pH scale here, and suddenly the blood has gone to the acidic side. It's no longer just a tad bit more than 7. It's less than 7. Then a buffer actually absorbs those extra hydrogen ions, because remember, acidic means that you have more hydrogen ions running around. And so because of this buffer, your pH level returns to normal. So you're no longer acidic because the buffer actually absorbed the extra hydrogen ions. And when the blood pH level becomes too basic, when we look here at the pH scale again, here's 7 in the middle, we've moved toward the basic side, then a buffer can actually release hydrogen ions in order to bring the blood pH back down to normal. And so this is a safety measure that our bodies actually have in order to control pH. And let me tell you how critical that is. That if you were to reach, say, a blood pH of 6, you would die. And so that's why your body actually has buffers. If you were to reach a blood pH of, say, 9, you would die. And that's why your body has buffers. Okay? And buffers basically help to bring the pH back into a normal range. Okay? But let's go ahead and start talking more about macromolecules. Macro meaning large, right? And with this, we're going to start talking about the chemistry of the foods that we eat. So let's talk about carbohydrates first, because they're very important. Our bodies actually love carbohydrates. And that's because we can get down to this little molecule that you're going to become very familiar with, which is glucose. And glucose has the molecular formula of C6H12O6. That means that there's 6 carbons, 12 hydrogens, and 6 oxygens. Okay? And this is the molecular configuration of glucose. And you're going to see that conformation is important, right? I think I'd spoken before when we were talking about atoms that when we're creating molecules, the way these basic carbon, hydrogen, and oxygen molecules actually come together to make a functional molecule is important. And it's much like origami, where you can start with this blank piece of paper folded in one way, it's an elephant folded in another way, it's a swan, right? It's the same piece of paper. It's just like that with molecules. So the way and the shape of the molecules is very important. So let's talk about glucose. So the carbon-hydrogen bonds of carbohydrates store a great deal of potential energy, and they're easily broken down by organisms such as us. And that's why they're used and preferred by organisms, right? Because we can easily break that carbon-hydrogen bond to get energy. And we're going to talk more about energy later, but the way I want you to think about it is when you break a bond, you get energy, you release energy. And a nice visual that I like to use is, have you ever gone camping and you have those glow sticks and then you crack them and then you see light? That's a chemical reaction. But think of that. When I break that bond, I'm getting energy, right? Just like a glow stick. When I crack it, I get energy. So it's the same thing for organisms. When they break the bond between this carbon and hydrogen, energy is released, and we need that energy in order to function. And so carbohydrates, such as glucose, are basically composed primarily of three elements: carbon, hydrogen, and oxygen. And carbohydrates are the primary fuel source for cellular mechanisms. Okay. Let's look a little bit closer at how conformation matters. So here's some common monosaccharides. And again, we're going to get into terminology, right? We talked about macromolecules, larger molecules. Now we have monosaccharides. Mono meaning one, saccharide meaning sugar. So I want you to be able to learn what these prefixes and suffixes mean in the Greek and the Latin, because that way when you come across a word that you've never seen before, a scientific word, the majority of the time you'll be able to figure out what that word means just by looking at the prefixes and the suffixes and understanding what they actually mean. So mono means one, saccharide means sugar. So we're going to look at some common monosaccharides. So we already looked at glucose, and here we have glucose here, C6H12O6, and we see the molecular conformation. But here we have fructose, and fructose also has the molecular formula of C6H12O6, but the conformation is different. So look here, and let's just start at this oxygen that we have here. Take any oxygen here and follow it, and you'll see that we have a very different conformation as we look at glucose versus fructose, even though the molecular formula is exactly the same. Same thing when we look at galactose, exact same molecular formula, C6H12O6, but it's the conformation of those molecules that makes a completely different molecule, and it's going to function differently in the body because of that conformation. Okay? So let's talk about glucose, our C6H12O6, in your blood. So glucose basically has three fates when you take in glucose. Okay? So first off, say you actually need the energy right away. You have taken in a high glucose meal and you need to run a marathon, right? You're going to start burning that energy. So the molecular bonds are broken down and new lower energy bonds are formed, and the fuel for cellular activity, that energy that you got from breaking those bonds, is now used in order to run that marathon. Okay? But if you have a meal that's high in glucose and that energy is not needed, it has two potential fates. So first, it can be stored as short-term energy storage as glycogen. Okay? So it can be stored as this molecule called glycogen. And glycogen is stored in your liver. Okay? And that's short-term storage. Easy to get to, right? It's kind of like storing things in your garage. Very easy to get to. I can just open up the garage door, boom, I can get to it, pull out the glycogen, start breaking it down, get to the energy that I need. Short-term energy storage. Or the body can put it into long-term energy storage. And to do this, it converts it into fat. And think of long-term energy storage, this fat storage, kind of like putting it into a storage facility that's far away. It's going to take a little bit longer to get to that energy, but it's there, right? And you can store a lot of it. Okay? So, three fates for glucose when you take it in. One, it can be used right away. Bonds broken, energy released, used right away by the cells. Or the energy is not needed. You had a really high glucose meal and then you lay down to take a nap, your body is going to either put it into short-term storage as glycogen, or it's going to convert it into long-term storage as fat. And we see that knowledge being used by people who run marathons or have to do like a triathlon. Right before, like that morning and the evening before as well, they'll have a meal that has really high carbohydrates, like a pasta meal or a pasta dinner, so that they get this huge push of carbohydrates, right? That will be put into glycogen so they can tap into that during the marathon. Okay. But here's a question. So when you go on a diet, why is it that you lose water weight first when you diet, right? You go on a diet and you're so happy because immediately within a day or two, maybe even the first week of dieting, boom, you lost like 5 pounds, right? And you're super happy. And then all of a sudden, it's harder to lose that next 10 or 20. So why, and it's called water weight, why do you lose that water weight when you first start to diet? Let's take a look at that. So it's really, really cool. I told you about glycogen, right? That's the short-term energy storage method that the body uses to store those carbohydrates. But it turns out that the body actually wraps those glycogen molecules in water. So a pound of glycogen has about 4 pounds of water molecules bound to it. So it's almost like your body is storing not only, an energy snack, but the water bottles to go with it, which is really cool when you think about it, right? So when you actually have to tap into that glycogen, you actually have to use that water that is surrounding the glycogen as well. And so that's why when you first start to diet, you're going to lose a lot of, quote, water weight, and you'll see the pounds just fly off, is because you're tapping into those short-term glycogen storage molecules that you have. They are wrapped in water, and you're going to have to use the water as well. But then once you've actually used the glycogen, well, now you're tapping into fat, and fat doesn't get wrapped in water. And it takes a little bit more effort to tap into the fat, okay? So more terminology. We talked about monosaccharides. Now we're going to take a look at polysaccharides and what they are, what that means. So poly means many, saccharide, sugar. So monosaccharides like glucose and fructose, if I chain them together, if I put them together, if I put 2 of them together, I've made a disaccharide. Di means 2, okay? So if I put 1 glucose and 1 fructose together, that's 2 molecules, I made a disaccharide. If I chain a bunch of them together, I've made a polysaccharide, many sugars, okay? So that's the terminology. Really super easy to understand. When you take a look at those prefixes, it tells you exactly what it means, right? So disaccharide is basically formed by the union of 2 simple sugars, 2 monosaccharides. And then complex carbohydrates are polysaccharides formed by the union of many simple sugars. So I chained a bunch of monosaccharides together, I make a polysaccharide, and we call that a complex carbohydrate. And when we talk about starch, this is just hundreds of glucose molecules as well. So digestion terminology. Digestion is when you're actually taking these bonds and breaking them down. So I can break the bonds between these 2 disaccharides, between glucose and fructose. And when I break a bond, I get energy, right? And that's the reason why I want to break those bonds. I want to get to that energy because my muscles need it. They need the energy in order to help me walk, help me run. When I break down complex carbohydrates, when I break down polysaccharides, I'm also getting energy, but I have to keep breaking them over and over to get to more energy, right? So between each bond, I'm getting energy, but it's going to take a bit to make my way through the entire chain of that polysaccharide, okay? So here's where that knowledge becomes important. So depending on their structure, dietary carbohydrates can lead to this quick but brief energy boost, or you can have this slow but persistent increase in blood sugar. So let's take a look at this graph that we have here. So on our y-axis, we have the blood sugar level, and on the x-axis, we have time. So when we take a look at fructose, and I'm breaking those bonds between the carbon and the hydrogen, I can get to that energy really fast, really quick, right? But it's single molecules of fructose. So I'm going to get to the energy really fast, and over time, I'm going to run out of fructose molecules to break down, right? And so the blood sugar levels are going to start to go down. So I get this high spike, and then it starts to go down pretty quickly. And that's drinking something like orange juice, which is basically has quite a bit of fructose in it. However, if I eat something that's a complex carbohydrate, like this bowl of oatmeal, right? It's filled with polysaccharides. It's going to take me longer to break my way through those bonds, but I've got more of those bonds to continue to break and use that energy over time. So I'm going to get this slow but persistent increase in my blood sugar. Okay? And so here's the question for you. Now knowing what you know, knowing how monosaccharides work, how complex carbohydrates work, how polysaccharides work, why we actually want to consume them, what is the purpose of us consuming them. If you have someone who is a diabetic and they had an insulin injection, but they didn't eat, right? Blood sugar is now very, very low, dangerously low. And this person starts to get weak and wobbly because they don't have enough bonds to break, right? Which are you going to give them? Because they're about to pass out. Are you going to give them a molecule that has fructose, a monosaccharide, or are you going to give them a complex carbohydrate? They're about to pass out, right? So here's the answer. You're going to give them a monosaccharide. You're going to give them the fructose. You're going to give them a glass of orange juice because you want that quick rise in their blood sugar level because you don't want them to pass out, right? And then you would follow that, once they feel better, with a bowl of oatmeal, right? Something that's going to give this slow, steady, continually present rise in the blood sugar level, right? Because if you were to give them the complex carbohydrate, such as oatmeal, you're not going to get a rise in their blood sugar level fast enough to prevent them from passing out, as you can see here by the graph, right? And so this is why you commonly hear when someone has passed out on the floor to put like a piece of candy in their mouth, or more commonly we'll use some Karo syrup, rub it on the gum so that it's quickly absorbed, and basically that blood sugar comes up very quickly. It's surprisingly fast. But if they've passed out, you certainly don't want to pour orange juice into their mouth because they're going to choke, right? And that's why Karo syrup or even a piece of candy can be used in an emergency. But that's why we reach for that, okay? So let's carry on. So We want to take a look at other ways that nature uses sugars to build strength in materials. And these things you're actually familiar with, but we're going to put a scientific name to them. So if you've ever gone to like a lobster house or Joe's Crab Shack and eaten lobster and crab, you know that it is really hard to get into that shell. Well, that shell is made of chitin, and chitin contains hundreds or even thousands of sugar units linked in as cellulose. And in chitin, the units actually contain nitrogen, and they have this increased hydrogen bonding within and between the chains, and that's what's giving the strength. And that's chitin, and it's very strong, as you can relate to if you've ever tried to crack into a crab or a lobster. That shell is made of chitin. When we look at cellulose, this is what gives trees their strength. Cellulose is actually chains of hundreds or even thousands of linked glucose units with hydrogen bonding between these parallel chains. And this contributes to trees' rigidity and makes wood this very desirable building material. So it's this linked bunch of glucose units. Okay. Let's talk about cellulose, because you've heard of that. And if you get a product such as Benefiber or some of these high fiber nutritional supplements, what's in it is actually cellulose. Cellulose is fiber. It's indigestible. We can't digest it, but it still aids in digestion, and it has a bunch of health benefits, but it's also helping to feed the microbiome that's in our digestive tract, that's in our gut. And it turns out, and science has just learned this recently, it turns out that feeding and taking care of the microbiome is extremely important to our health, because this bacteria, and we're going to talk more about this later as we get into learning about cells and bacteria, but the bacteria that's in our gut, they're doing a lot of work for us. They're actually modulating our immune system. They're helping to keep us healthy, right? And so when we take cellulose into our diet, we're actually helping to feed them. And that's why we have better digestion, right? We're moving a lot of the food and the waste throughout our GI tract in addition to feeding our gut microbiome. And so cellulose in our diet is very important, and that's fiber. But now I want to talk a little bit more about a different macromolecule, and that's lipids. So we kind of opened up the door to lipids, and we just kind of talked about them very briefly earlier. But let's take a look at this emperor penguin here. They actually benefit from the insulating effect of fat, and they're able to thrive in the extreme cold and temperatures of places like Antarctica because of fat. Fat is not bad. I know if you're from California and you live in California and you were raised in California, you probably feel that fat is a really, really bad thing. Turns out it's not. So let's talk about some features of lipids, also known as fats, right? There's different types of lipids, but they have certain features. So number one, lipids are molecules that do not dissolve in water. You put lipids in water and they do not dissolve. They're very greasy to the touch. And they can be a significant source of energy storage. That's why our body actually will convert those excess carbohydrates into fat, right? So 3 basic types of lipids that I need you to know. One is fat, right? Main function is long-term energy storage and insulation. But then we also have sterols, right? And their function is to regulate growth and development. And then we have phospholipids. These are extremely important. Every cell of your body, the cell membrane, is made of phospholipids. And if you didn't take any lipids in, if you didn't create any lipids, then all of your cellular membranes would break down. So lipids are important, right? When we think about sterols, if you're not familiar with that, that's like your hormones, estrogen, testosterone, thyroid hormone. These are all sterols, okay? So lipids are important. But as in the other molecules that we talked about, it's all about conformation. So let's take a look at the structure of fats, triglycerides. So fats are composed of primarily 3 elements: carbon, hydrogen, and oxygen. But again, it's the conformation, right? And the reason why our body uses fats is because this is a lot of high energy bonds. Remember that carbon-hydrogen bond that we can break to get energy? Well, take a look at this fatty acid tail. Look at how many carbon-hydrogen bonds we have to break there. How much energy is stored there, okay? That's a lot. Now, fats have a common conformation, so they have a head. Here, the head is glycerol, and then they have a tail. And these are the fatty acid tails that we see here. This is looking at fats on a molecular level, okay? And so not only do we store our excess energy that we've taken in the form of fats, but we also have evolved to want to take in foods that are high in fat because we want to get to these energy bonds, right? And so when we look at something like this plate that is brought to us, or these plates, right? One that has this creamy gravy and these eggs and hash browns and bacon and toast with butter on it, we're like, yum, right? Because that's a lot of fat. And we have evolved to actually want to eat things like that because we've evolved to expend a lot of energy throughout the day, and you don't really know where your next food source is going to come from. And so you have an inclination to want to eat these high fat foods because that's a lot of energy in those fat bonds, right? However, in our current day and age, we don't expend a lot of energy in our daily lives, right? We're not out working on the farm to grow the food. We're not, taking long walks to actually go and harvest from the forest. There's not a lot of energy expenditure happening in our day-to-day lives, but we still have that craving for high-fat foods. And when we give in to that, then that's when we can have a problem with obesity. So don't feel bad when your body is actually looking at these foods and wanting them. So is every human on the planet, because that's what we've evolved to do. However, there's a reason for it. All right, let's carry on and take a look at some more terminology so you can understand. You've probably heard these terms, saturated fats versus unsaturated fats. What does that mean, right? Let's take a look. So in saturated fats, each carbon in the hydrocarbon chain is bound to 2 hydrogen atoms. Can you see this? So here we have, in this illustration, we've got the black balls, which are the carbon, and then we've got 2 hydrogen atoms that are bonded to each carbon. What this does is it gives this straight configuration to the fat molecule. And so these straight fatty acid chains can be packed together very tightly. Think of it as like the combs of a comb. When they're straight, I can put a lot of those combs together in one spot, right? So as a result, these are called saturated because there is a hydrogen on every, 2 hydrogens on every carbon. The carbons are saturated. So as a result, saturated fats are solid at room temperature. So if you're trying to figure out if the food that you're about to put into your mouth is a saturated fat or an unsaturated fat, a good way to determine that is if it's solid at room temperature, it's probably a saturated fat. So that stick of butter, solid at room temperature, saturated fat, right? Olive oil at room temperature, it's liquid. It's an unsaturated fat. So saturated fats, straight fatty chains, I can pack more into one space, right? But let's look at unsaturated fats. Unsaturated fats, now what we have is at least one carbon in this hydrocarbon chain is bound to one hydrogen. And so we have a double bond here between the two carbons. And see how this one carbon only has one hydrogen? Well, that single hydrogen and this double bond results in this conformation where it has sort of a kink to it, which means I can't pack as many into one space. And that's a good thing, right? So this crooked conformation of the fatty acid chains makes a difference. And it's all because the carbon, at least one carbon in the chain has only one hydrogen in this double bond. Now, crooked fatty acids can't be packed together tightly, and as a result, unsaturated fats on their own, such as olive oil, are liquid at room temperature. Well, let's talk about something else another term that you've probably heard, and that's hydrogenation. Right? So hydrogenation can improve a food's taste and texture, shelf life, but it also makes the food less healthy. Let's look at why. So hydrogenation is actually the artificial addition of hydrogen atoms to an unsaturated fat. So here I've got this unsaturated fat, and it's unsaturated because I only have one hydrogen on my carbon, and I have this double bond here. It's giving me this nice crooked conformation. I can't pack a bunch in. That's basically going to clog up my arteries. But through chemical means, I can add more hydrogen atoms to an unsaturated fat and do what's called partial hydrogenation. You can see here where some of those carbons that did have just a single bond, now another hydrogen was added on, right? Suddenly I'm getting a straighter conformation to my once unsaturated fatty acid conformation, nice crooked conformation. Now when it's hydrogenated, it's becoming more and more straight, right? So this partially hydrogenated food is more likely to be stored as fat, right? So you probably wonder, well, when I told you that you had a couple of fates of glucose, it could be stored as glycogen or it could be stored as fat. The question that would arise is how does the body know which one it's going to choose, right? Well, some of that choice depends on the conformation. So these saturated fats or these partially hydrogenated fats that are made to look conformationally more like a saturated fat are more likely to be stored as fat in the body, and that can be a problem, right? All right, so sterols. Let's take a look at sterols. So I told you that an example of some sterols would be estrogen and testosterone, because you're very familiar with those. Those are hormones. They're also called steroid hormones, and they can regulate sexual development, maturation, sex cell production. Estrogen influences memory and mood. Testosterone stimulates muscle growth. And so you're very familiar with these steroid hormones, these sterols. But one that I want to introduce you to that you may not be quite so familiar with is cholesterol, which also has gotten a very bad rap. You cannot eliminate all cholesterol from your diet because it's important. And that's because cholesterol is actually, again, part of our cell membranes. We use cholesterol as a very important component of our cell membranes. And so dietary cholesterol can attach to and thicken vessel walls and may cause serious health problems. So we want to limit how much dietary cholesterol we take in, but you cannot eliminate it in its totality because we need it to actually form our cell membranes. So all sterols are based on a structure featuring 4 fused carbon rings. So they look very similar to this. Okay. What I want you to remember when I say sterols, I'm talking cholesterol, I'm talking your steroid hormones, your estrogen, your testosterone. Okay. But I want to take a closer look at phospholipids. Remember I told you that phospholipids make up our cellular membranes. Every cell in our body, the cell membrane, is made of phospholipids. So I want to look closer at that. So here we have 2 parts to a phospholipid. It has this phosphate group, hence the name. And we have these carbons, and we have a nitrogen, and we have all of these hydrogens. And this conformation that we see here, this is called a hydrophilic head. Hydro meaning water, philic meaning loving, so water-loving head. This portion of a phospholipid is actually attracted to water. So this hydrophilic head is attracted to water. Then as we come down, we have the fatty acid tail, and the fatty acid tails are actually hydrophobic. So hydro, again, meaning water, phobic, meaning not liking, repulsed by, okay? So these hydrophobic tails, they are not attracted to water. They want nothing to do with water. Now, why is this important that we have this hydrophilic head and this hydrophobic tail making up these phospholipids? It turns out that when you put phospholipids in water, it will automatically align itself to form a membrane based on the fact that the hydrophilic head loves water, so you're going to get an orientation of the head towards water, and the hydrophobic tails hate water, and so they're going to align away from the water, and they're going to make these instant membranes. This is almost like what's called click chemistry, which is really cool. And so you'll see this symbol used to represent phospholipids, but what I really, really want you to remember, and you will be asked on an exam, is how these portions of the phospholipids, the hydrophilic head and the hydrophobic tail, which portion likes water? The hydrophilic head likes water. Which portion hates water? The hydrophobic tail hates water. And so when you look at the cell membrane, you're going to see this click chemistry, this conformation that makes the cell membrane possible because of either the attraction or repulsion of water to phospholipids. So phospholipids, very important. Remember what hydrophilic means. Remember what hydrophobic means, okay? And the alignment that we see in the head and the tail in relationship to water, okay? These guys are just so ugly, they're cute. All right, let's talk about proteins. So proteins are a huge group. We're carrying on with our macromolecules, right? So proteins perform a huge variety of different functions. They all, however, are built pretty much the same way and from the same raw materials in organisms. So proteins make up both structure, protection, regulation, contraction, and transportation functions in the body. So Structure. When we think about proteins that form structure, we think about hair, fingernails, feather, horns, cartilage, tendons. An example here is the feather of a scarlet macaw. When we think about protection, proteins are part of the group that help to fight invading microorganisms. They help to coagulate blood. Here in this image, you're seeing a blood clot. We can see the red blood cells. We can see a white blood cell. We can see all the fibrin threads here. When we think about regulation, proteins are helping to control cell activity, and they make some of the hormones as well. They comprise some of the hormones. So here we're looking at a goblet cell in pink and blue, as you can see here, in the mucosal lining of the small intestine. So here we have the small intestine and we have a goblet cell, and it's making mucus. That's protein. Proteins are involved in contraction. So proteins actually allow muscles to contract, the heart to pump, sperm to swim. So here we have heart muscle cells made of protein, right? Protein in transportation. So proteins actually can carry molecules such as oxygen around the body. And we'll talk about what that looks like, why we actually breathe, and the molecules that are actually making that happen. But proteins are a huge part of that, okay? But what is a protein? So let's take a look at it. So proteins are actually made up of amino acids. And this is what an amino acid looks like on the molecular level. So let's break it down. So here you have what's called the amino group. It's a nitrogen. And here we have the hydrogens that are actually bonded to the nitrogen. And then we have what's called a carboxyl group. And here we can see oxygen and carbon. And so we always have this amino group and this carboxyl group in a protein. What's different and what makes different amino acids is what's in this side chain. So for example, if I were to attach in this side chain this molecule here, which is a carbon and 1, 2, 3 hydrogens, I attach this carbon and 3 hydrogens onto this side chain right here, just stick it in as the side chain, and suddenly I've made alanine. Or if I were to stick as this side chain 1 hydrogen, just click the hydrogen on there, now I've made glycine. Change the side chain to this molecule here, tryptophan, this molecule, and I've made tryptophan, right? So the amino group is always the same. The carboxyl group is always the same. It's the side chain that's changing in order to make a different amino acid. So it's the side chain that's the unique part of each of the 20 amino acids, causing this variation in size, shape, and charge. So the side chain is the important factor here in amino acids that gives us the different amino acids. So why do we care about different amino acids? Well, because we need to build all those proteins that we just talked about to make our hair, fingernails, part of our immune system, right? We need these proteins. And so let's look at some of these proteins. Just give you some of these names. We have histidine, isoleucine, leucine, lysine, methionine and cysteine, phenylalanine and tyrosine, threonine, tryptophan, and valine. So you'll hear the terminology complete versus incomplete proteins. Well, what does that mean? So let's look at some foods. We have here basically on what looks to be our x-axis. So we have apples and almonds and white rice and lentils and milk and tofu and chicken and beef. And we can look at the constituents of the amino acids that these foods are made up of. So the solid dot means that it has that amino acid, and the open dot means that it's missing that amino acid. So when we look at apples, we can see they're missing isoleucine. Apples are missing methionine, cysteine. They're missing phenylalanine and tyrosine. They're missing threonine. They're missing tryptophan. These are all required by us in order to live and make all the proteins that we need to function. All of these amino acids are required. So if I were to try to live on a diet of apples, I would soon become deficient, right? That's why you can't live on just apples alone, right? Because we're missing these amino acids that we very much need. Same thing with almonds. We can see that we're missing lysine, we're missing methionine, and we're missing cysteine. But let's take a look at a very common combination, and let's understand why it's such a common combination. Let's take a look at white rice and lentils, beans of some sort, right? So when we look at white rice, we can see that it's missing lysine. But when I look at lentils, I can see they have lysine, but they're missing methionine and cysteine. But the rice has methionine and cysteine. So if I eat a combination of white rice and lentils, I'm actually getting my full complement of amino acids that I need to build the proteins that I need to make. And that's why beans and rice are such a common food staple throughout the world. They may come in different names and different forms. The lentils themselves may have different names. The rice may be a different type of rice. However, by taking these incomplete proteins and pairing them together, I can actually get a full complement of the proteins that are required. Okay, let's look at some complete proteins. Here we have milk, it's complete protein. Here we have tofu, it's a complete protein. Chicken, complete protein. Beef, complete protein. So in order to get my full complement of proteins, without having to do this mix and match, I can eat beef, I can eat chicken, I can eat tofu, I can have a glass of 2.5% fat milk as well and get the full complement of proteins. But I can also get my full complement of proteins by eating the combination of rice and lentils. Okay, so remember that. That's been known to be asked on the exam. And that's why we see rice and lentils as traditional dishes in so many cultures around the world, on this combination of different incomplete proteins coming together to provide all the essential amino acids. Okay. But I want to take a look and show you more about the structure of proteins, because this becomes very, very important. And remember this, the fact that conformation is really, really important. We're going to see this play a part when we start talking about viruses and antibodies and that sort of thing, that conformation is extremely important. So let's understand the conformation of proteins. So here we have our amino acids that we saw before, right? And these amino acids come together, they bond together using these peptide bonds. It's a new bond between different amino acids. And so this is called the primary structure, where we're just looking at the amino acids bonded together. But from there, we start to get these hydrogen bonds that start to form these corkscrew-like twists and these pleated folds. And this is called the secondary structure. So suddenly we start taking these chains of amino acids, and then through this additional bonding, these hydrogen bonds, we start to change the conformation of the protein. So we get this secondary structure. And then from there, we continue to shape and fold, and we get this complex three-dimensional shape that begins to form. And it's formed by these multiple twists and bends that are happening in the protein structure in this polypeptide. And this is based on the side chains and different bonding interactions that are happening. Also, what's happening in the liquid that this protein is actually in. We can get changes to the structure. So now we're looking at what's called a tertiary structure. So we're now getting into a higher three-dimensional structure. And then we can move on to what's called a quaternary structure, where now we can have two or more polypeptide chains bonded together. So now we're starting to see more bonding of the different polypeptides, okay? So protein structure is extremely important because protein misfolding, the fact that a protein actually has been folded in the wrong way, actually can cause disease. And there are certain diseases that are based upon protein misfolding. Okay, so if you want to learn more or have interest in that, all you have to do is Google diseases due to protein misfolding. And you'll see that there's quite a few diseases that actually are caused by our proteins being misfolded. And so in the portion of the cell that makes our proteins, and we'll get to that in weeks that are coming up, when we talk about the areas of the cell that actually are making the proteins, there's checks and balances to make sure that the new proteins that we made are actually folded properly. The cell checks that before it sends the protein out. But this knowledge of protein folding, we actually use, right? So it turns out that here you have a normal protein, but I can disrupt this protein. I can do what's called denaturing this protein, which means that now this protein doesn't work. If I unfold, if I denature a protein, that protein will no longer function. And I can denature a protein through extreme environments. The most common is extreme temperature, which means high temperature, heat, right? I can use extremes of pH, so I can use strong acids, I can use strong bases. These will denature a protein. And so they're going to disrupt the protein shape and thereby disrupt a protein function. And we use this knowledge every day. You use this knowledge, the knowledge of denaturing proteins every day without even thinking about it, right? So I want you to take a minute and just think about where in your world, in your life, in your day-to-day life, are you actually using the knowledge of extremes of environment to denature protein? Take a second to think about that. So it turns out when we put foods in the refrigerator to keep them cold, to delay the foods decaying, we're actually disrupting the proteins. We're slowing enzyme action, and enzymes are very specific, very specialized proteins. We're slowing down their action in the microbes that actually would cause the destruction of our food, right? Because we put it in the refrigerator, we slowed the enzyme action of those microbes. But if we want to stop the action of those microbes, what do? We cook the food, extreme temperatures. We put it in a hot pan, right? And we cook it, completely going to denature the proteins of the bacteria that are there, right? That's what makes our food safe to eat. So other ways that we actually use this knowledge of denaturing proteins is when we want to change the shape of our hair, right? We will flat iron the hair, and that will not completely denature the protein, because then it would just melt away. And you might have done that in your younger days when you put a curling iron on and left it too long, take it off, and there's your hair in the curling iron, right? You completely denatured that protein. But what we do is we can partially denature it to straighten it out, right? Or we can turn it onto the curling iron and change the structure. And when you pull it off, partially denaturing it, now you have curls. We can do it on a semi-permanent basis, right? By using exactly what's called perms, permanent. Usually lye is used. That is the strong pH change that we're using to partially denature the protein. The same thing, If you totally denature the protein and leave it on too long, your hair is just going to melt right out, right? But we can use that to denaturing of the protein of the hair to either make the hair straight or to make the hair curly. So we use that in our fashion as well, right? So I'm going to stop right there. And that's the end of our lecture for this week on pH and macromolecules. So make sure you understand all that terminology that was part of this lecture and this PowerPoint. It's extremely important. We build upon that knowledge moving forward as we continue on through the course. Thanks for joining me. I'll see you in the next lecture. Bye.